mRNA vaccines haven't been around for very long, with most development only occurring within the last 30 years. Vaccines generally work all in the same way, to train the immune system to recognize and attack viruses before contracting the illness. Some vaccines contain small doses of the virus itself and are injected into our body. However, mRNA vaccines are unique because they don't inject the virus themselves, but only the mRNA segments, so our cells can build parts of the virus themselves. Think about other vaccines as delivery men who drop off a completely assembled piece of IKEA furniture, whereas mRNA vaccines only deliver the instructions manual, so all the materials need to be supplied yourself. In this video, we will be delving into 4 main parts: how mRNA vaccines are designed and structured, how mRNA vaccines are delivered into our cells, the translation of mRNA vaccines into COVID-19 spike proteins, and how mRNA vaccines trigger an immune system response. We will be looking specifically at COVID-19 and how mRNA vaccines help protect us from this highly infectious virus.
How mRNA vaccines are designed and structured. Unlike the conventional vaccines, which contain a suspension of dead or attenuated pathogens, mRNA vaccines utilize certain component of the pathogen, the antigen protein. In essence, mRNA vaccines are made up of a solution of messenger RNA strands that encode for the specific antigen of the pathogen being targeted against for immunization, thus allowing for immunity whilst reducing major side effects. Think of it as being able to eat a whole cake without letting the sugar and the carbs get the best of you. To avoid any errors during the translation phase, which we'll cover soon, these mRNA strands are modified on each end. The 5' end is capped off by a modified guanine base, while the 3' end contains a poly-A tail, a chain of consecutive adenine bases. This modification ensures that the mRNA strands are less likely to be damaged during the translation process. The introns in the mRNA strands are preemptively spliced to remove any nonsense genetic codes. When an mRNA vaccine is injected, it invades the body's cells and initiates the body's immunity in the same way our body cells fight off any other pathogen. Firstly, the mRNA transcriptase from the vaccine is attached to the ribosome inside the cytoplasm of each invaded cell. The ribosome then reads the code the mRNA carries to produce a chain of amino acids. After this, transfer RNA molecules attach to the mRNA strand and read the bases 3 at a time, producing the corresponding amino acid for each triplet coded. Resultantly, as the mRNA is completely translated, a new protein is produced, and in this case it would be the antigen of the targeted pathogen. In other words, this protein would be the cake without the sugar and the carbs.
A conventional vaccine contains ORF for the target antigen, flanked by ultra-translated regions called as UTRs, with the terminal poly-A tail. Another kind of mRNA vaccine is based on the genome of positive strand. Vaccines made by this type of preparation are called self-amplifying, as they can self-replicate. By using the enzyme RNA-dependent RNA polymerase, it generates multiple copies once it is injected.
mRNA, which is the code-carrying molecules produced in the body and function as an instruction in the protein production. If mRNA could be designed and used intentionally to reach the ribosome, cells and the immune system could be informed about how to fight targeted virus. To help design mRNA delivery, lipid nanoparticle or nano in the short term is required, which are small and fatty droplets that can carry mRNA safely as a container. After mRNA successfully arrives at its destination, the nano will disappear naturally. At the end of the transport process, mRNA inside the fatty vesicle will be taken by the dendritic cell, which are related to the immune system. While joining the dendritic cells, mRNA will stay in the cytoplasm. Then those mRNA will create an enzyme that the human body needs, and be observed by ribosomes. After those ribosomes read mRNA, the ribosome will be capable to produce the specialized protein that can fight against the virus.
After the mRNA enters the cell, the mRNA is translated into the COVID-19 spike protein. This spike protein is only a specific segment of the viral molecule, not the entire COVID-19 virus itself. This spike protein is chosen because this unique shape is the mechanism by which COVID-19 virus attaches to our cells. So, noting that an mRNA is just a copy of COVID-19's DNA, how can the instructions carried lead to the production of a spike protein? This process is called protein synthesis. The mRNA first escapes its lipid nanoparticle protective shell and moves into the cytoplasm, where it attaches to a ribosome. Transfer RNA molecules, tRNA, from the cytoplasm carry amino acids to the ribosome. As we know, amino acids are the building blocks of proteins, which are then strung together to create our spike protein. Our mRNA directs which tRNAs come in and in what order. It does this as mRNA contains codons, which are triplets of nucleotide bases, A, U, C, or G in RNA. tRNA contain anticodons that try to find the complementary bases on the mRNA, and once found, they transfer their amino acid. There are 20 amino acids in total, and each codon corresponds with an amino acid. We can refer to an mRNA codon chart to tell us which bases match up to which amino acids. These amino acids are connected together with peptide bonds until it reaches a stop codon, which tells the ribosome that protein building is finished. Once this translation process is finished, voila! A spike protein is produced and transported outside the cells so that our immune system can start to familiarize themselves with this spike protein, which we will talk more about in the next stage, how mRNA vaccines trigger an immune system response.
Vaccines are laboratory preparations of biological substances which are injected in order to provide protection against diseases. It provides active acquired immunity. Typically, a vaccine is made by a dead or weakened organism of that particular disease from which we want protection. Once the weakened organism enters our body, in response to the antigen, our body creates antibodies. In the future, when the actual organism enters our body is already prepared with antibodies to fight. Apart from these traditional vaccines, professionals have curated messenger RNA vaccines. Messenger RNA vaccine codes for a protein which is present in the membrane of pathogen. Usually, a critical protein is chosen. So unlike the traditional vaccines, instead of pathogen being injected, only its genetic material is injected. When these vaccines are injected in the body, muscle starts to translate them and produce protein. When the protein is formed, as a part of our normal immune response, antibodies are formed, which in future protect from the actual pathogen.
The immune system reacts to the infection of the pathogen and uses adaptive immunity to fight off any antigens in the body. Although it may be a slow process, even the initial mRNA transcriptase from the vaccine is eventually destroyed by the body's immune system, resulting in immunity through memory B cells. Furthermore, using the body as a factory for the production of these antigens allows the cells to replicate and translate the protein encoded by the mRNA into multiple copies, therefore proving more effective in building immunity even in small doses of the vaccine.
So, in short, mRNA vaccines are specifically designed and structured with specific antigens in mind, are carried in a lipid nanoparticle protective bubble, create proteins through translation, and induce antibodies to boost our immunity. I hope this has been educational for you all, and thank you for watching.